Welcome to the YouTube live classes for botany for class 12th. Now today we will go for another new topic of molecular biology, that is transcription and the post transcriptional modification. So what we are going to discuss now, what is transcription, how it happens in prokaryotes and in eukaryotes, and what are the different process or the phases of transcription like initiation, elongation and termination, and finally what happens to the RNA molecules after the process of transcription that is over. So before going to transcription, let me tell you about the central dogma of molecular biology. Now what we have studied in the last class is DNA replication. Now DNA replication is the process where DNA replicates itself or it synthesizes a new copy of itself, which will be the exact replica of the old one. Now as we know, now the DNA, it replicates to form the DNA. Well, but the most important thing is that now it has the capability to produce a copy of mRNA or any RNA. Okay. So it has the capability of producing RNA from its own copy. How, that we will discover today. And the RNA strand, and most likely the mRNA strand, it gives rise to a copy of polypeptide which undergo certain modification to form a functional protein. So what it tells, the central dogma of molecular biology says that from a DNA molecule always a RNA molecule is transcribed or it is produced, and from the RNA, particularly the mRNA, now the protein or polypeptide, they are formed, and the process of formation of RNA molecules from the DNA strands is called as transcription. Okay? And it goes in a single direction. Once the mRNA is formed, it goes to the formation of protein by a process called as translation. Okay? And this total process, that is from DNA to RNA, then from RNA to protein, is called as central dogma of molecular biology. And most of the organisms, they follow this direction of the metabolic activities or this molecular biology, that is from DNA to RNA, then from to protein. But there are certain not exactly organisms, there are certain virus which actually has RNA as their genetic material, and it gives rise to the DNA, a complementary DNA copy, which again gives rise to the RNA copy and then to protein. And this process of formation of a DNA molecule from an mRNA or from an RNA molecule is called as reverse transcription. This is not the part of the central dogma, rather this is the reversal of central dogma which happens in virus. But let us not talk about the virus, rather let us talk about all the organisms which are included in the Whittaker 5 kingdom system of classification, or even you can take the 7 kingdom system of classification or even higher. So any organism starting from Monera or bacteria to the higher evolved organisms like human, now we follow this central dogma of molecular biology, which says that a DNA copy or a DNA molecule produces an RNA copy and a RNA molecule produces a protein by the process of transcription and translation respectively, and this is called as central dogma of molecular biology. So before going to the RNA transcription or the transcription process, so what is transcription? Transcription is the process in which a molecule or a segment of RNA is synthesized newly from a DNA segment. Okay. Now before going to that, it is very essential to know the structure of RNA. Now what is the RNA? RNA is also a polynucleotide. This is also called as a nucleic acid, but this is RNA, that is ribonucleic acid. We already have discussed the same structure of DNA RNA before, just a small reminder. So, the RNA molecule is nothing but this is a polymer of ribonucleotide. So, this is a polymer of ribonucleotide. So, one after another the nucleotides are joined together. So, this actually the structure of a RNA molecule. Now, here you see this is not double stranded, no RNA is double stranded, only double stranded nucleic acid or molecule is DNA. Okay. So, DNA is the only nucleic acid which is double stranded, rather no RNA is what double stranded except some viruses. Okay. Let us forget about that. But when we compare or when we stick to the organism from bacteria to human beings, now the RNA molecules may it be mRNA, tRNA or rRNA, now they all are single stranded molecules. Okay. So, this is one ribonucleotide, this is another like this, and they are being joined together by the same phosphodiester bond, as we have already studied the detailed structure of DNA and RNA. Now, this is single stranded molecule and the bases are present here like adenine, uracil, guanine, cytosine, guanine, uracil. Now, here you see what is the difference. In case of DNA there would be T here, and in case of RNA there would be U. So, no T is present in RNA and no U is present in DNA. So, this is the overall structure we already have discussed. So, there are three types of RNA, one is mRNA, tRNA and rRNA. Okay. Let us have a look on the structure. Now, the chemical structure is the same, the simple a single stranded molecule starting from the 5 prime phosphate group and ending at a 3 prime hydroxylic group. Okay. So, this is a 3 prime phosphate group and this is a this is a 5 prime phosphate group and this is a 3 prime hydroxylic group. If you go for a electron micrographic study, now you will see the physical structure as this. Okay. So, this will look like this, and what are this introduced or this structures, these are nothing but the protubed bases. Okay. So, this is the structure of mRNA. We have to know the structure of mRNA, tRNA and rRNA because this is very essential to know the structure in order to go for the process of translation, which we will discuss in the next week or in the next class. So, this is the structure of tRNA. Now, all the RNA molecules I have told you, now 3 RNAs are there, mRNA, tRNA and rRNA, and all the these three are involved in protein synthesis. Now, how protein is important for us, that we know it very well because proteins are the building blocks. Okay. And this all 3 RNAs are involved. mRNA forms the template for the synthesis of protein. tRNA brings the amino acid, it brings the amino acid to the site of protein synthesis, and rRNA is a part of ribosome that catalyzes the protein synthesis. Okay. So, all 3 are involved. So, the structure of all the 3 we should know. So, what happens? The template mRNA acts as a template in protein synthesis, tRNA brings or it pulls or it picks the amino acid from the cytoplasmic pool and it brings it to the template of mRNA for joining of the amino acids with each other, forming a polypeptide chain, and the ribosome in itself is the protein factory where the proteins are synthesized, and it provides the environment for the catalysis of that peptide bond and it forms the protein. So, this is the structure of mRNA, then is the structure of tRNA. Now, the structure of tRNA, that means the tRNA is synthesized as a single stranded RNA, okay, like any RNA from 5 prime to 3 prime. Later on, it undergoes the formation of a secondary structure Okay. So, the secondary structure says that there are 3 loops or in total 4 loops, somewhere another loop is not there. So, what are the loops? Let us see. Now, this is a single stranded molecule which after certain modification or after certain changes, it forms a structure like this, which is called as the secondary structure, and it is the clover leaf model of tRNA, or this is the clover leaf model of tRNA structure. Okay. So, it forms, so there are certain self complementary regions which as they are being self complementary, they form hydrogen bonds with each other, forming the stem regions. Now, you can see 4 stem regions are there. Okay. 4 stem regions are there from 5 prime to 3 prime. Then 3 loops are there. That means certain single stranded regions which is present within this clover leaf model structure or the secondary structure of tRNA. Now, these are called as D loop, anticodon loop, T loop, and this loop is called as the variable loop, and this is not being present in all the tRNAs. Somewhere it is present, somewhere it would not be present. So, anyways, now these 3 loops are important. One is T loop, another one is anticodon loop, another one is D loop. This T loop is conserved. This is a conserved sequence, and it is only having a T psi CG sequence which is present in any tRNA you take, irrespective of all the tRNAs. Now, this T loop is present and this has a conserved sequence called as T psi CG. And the anticodon loop, now it has 3 bases which forms the interlocking with the mRNA codon. Okay, that will discuss subsequently. So, the anticodons, they form up, let us suppose there is a there is a sequence here like UAC. Now, the codon sequence will be like this will be AUZ. Okay. So, there will be a what kind of hydrogen bond formation between the codon of the mRNA strand and the anticodon of the tRNA, and here the amino acid will be present at this point at the 3 prime end of the tRNA, and this also have a sequence called as CCA, and this A sequence carries the amino acid over here. Okay. So, this is the structure of tRNA, and the D loop, this has a kind of a kind of base known as dihydrouridine. Okay. So, this is the overall structure. Then from the secondary structure, a tertiary structure is formed, and this tertiary structure is a inverted L-shaped structure. This is a inverted L-shaped structure, you see. Okay. So, now you can visualize very well. This is a inverted L-shaped structure. So, this is the overall structure of tRNA. Now, you can see it here. Now, this this is the anticodon. When it folds further, now it forms this anticodon like this. So, these are the anticodon. This forms the interlocking with the codon. Now, you can see it here. Well, and the amino acid is carried at the 3 prime CCA end. Okay. And you see here the clear picture, this is an mRNA strand, these are 2 tRNA molecules, this is one tRNA, this is another tRNA. Now, you see one tRNA holds a polypeptide chain, another chain carries another new amino acid and it will be held here, or this one will be held here. Okay. And again this chain extends like this, and this is the ribosomes, well, this portion, and this ribosome slowly slides over this mRNA strand till it reaches the end point or the termination point. And how it slides? It slides once by covering 3 bases at a time, and these 3 bases or 3 nucleotides, they are called as codon. So, it slides over one codon at one time. Okay. So, this is the structure of tRNA, and the rRNA, this is also being what, synthesized as a single stranded molecule or RNA, but it undergoes the transformation into the secondary structure, and there is no such fixed structure for RNA. If this is the rRNA, then lots of ribosomal proteins, now they associate themselves over this rRNA. Okay. Now they associates themselves over the rRNA and they form what? This is the rRNA, this colored molecule, and this red dotted lines or this red dotted beads, okay, they are the R proteins, and togetherly the rRNA molecule and the R proteins, they form what? Ribosomes. And what it does, the ribosome is the protein factory, or this catalyzes the protein synthesis. Okay, so this catalyzes the protein synthesis. So these are the overall structure once again. Now RNA is of three types: mRNA, tRNA, rRNA. This is the structure of mRNA, tRNA and rRNA, and all the RNAs are involved in the formation or in the process of protein synthesis, which we will cover in the next chapter and the next week. So, in case of eukaryotes and prokaryotes, now the ribosomes, now they have what? The 80S ribosomes are present in eukaryotes, and the 70S ribosomes, they are present in prokaryotes. Okay, and they are being divided as 60S, 30S and 50S, 30S and 60S, 40S. Okay, and you see in the 60S, the larger ribosomal subunit of the eukaryote, now three short RNAs are there. One is a 28S, another one is 5.8S, and the last one is the 5S rRNA, which are present, and togetherly they form the larger ribosomal subunit of what? The eukaryotes or the 80S type. Then the 40S, it has a single rRNA which is of 80S type being combined with 33 proteins, and here all the three RNAs, they are combined with 50 numbers of proteins to form the larger ribosomal subunit, and here a single RNA which is a 80S rRNA, which is involved with the 33 proteins to form the smaller ribosomal subunit. Now next, in case of prokaryotes, similarly the 50S type of or the larger ribosomal subunit, it carries a 23S and a 5S rRNA, which are being involved with 34 R proteins, okay, to form the 50S type of ribosome or the larger ribosomal subunit. Then in case of 30S, there is a single rRNA component which is of 16S type, or this is a 16S rRNA component which is being involved with 21 proteins to form the 30S component of the ribosome or the smaller ribosomal subunit of the prokaryotes. Okay. So, this is all about the mRNA, tRNA and rRNA structure. Next we will go to, straight away we will go to transcription. Now what the transcription tells is the process of copying a segment of DNA. Now in case of replication, we also copied the DNA, but here a copy of DNA is what? Is a copy of DNA or a segment of DNA is copied into a segment of RNA, not into a segment of DNA, rather into a segment of RNA. So what it says is the process of copying a segment of DNA into RNA molecule. May it be this RNA can be a mRNA or a tRNA or a rRNA. Okay. So whatever it may be, only the mRNA copy, this will go for producing the protein in the process of translation, as we have discussed the central dogma of molecular biology. But the rest too, the tRNA and rRNA, they never produce any protein molecule, or they never act as a template for the synthesis of protein molecule. Okay. So, the segment of DNA transcribed into RNA molecules that encode a protein are said to produce mRNA, and the rest or other segments of DNA that are copied into RNA molecules only but not into protein, they are called as non-coding RNAs, and this is called as coding RNA or simply the mRNA, and these are the non-coding RNA. This can be tRNA, rRNA and many other RNAs are there which is not in our syllabus, like small nucleolar rRNAs and all. Okay. So, this is again a picture where you can see now the DNA molecule is being formed into a RNA molecule in the process of transcription. So, the process of transcription says that a segment of DNA is what copied into a segment of RNA molecules. It can be mRNA, tRNA or rRNA. So, what happens here, if we will go to the basic of this transcription and we will compare it with the replication system. Now, in case of replication also the whole DNA molecule here you can see now partly in form of replicons, okay, or in terms of replicating units. Now, they separate from each other, that means the double stranded molecule they are separated into single stranded template, then basing upon this 2 strand as template, now 2 new strands of DNA is synthesized. So, once the melting process starts, it goes on what enlarging the single stranded DNA template and simultaneously new double stranded DNAs will be created. Okay. So, out of which one will be old, another one will be new, but there in case of replication, now this is a permanent process. Once the DNA strands are separated from each other, now they will go on synthesizing a new copy of DNA. Whereas, in case of transcription, now what we are studying about? We are studying about transcription. Now, in case of transcription, now here the same melting process will occur, no doubt, okay. And here you can see that this is a this is a sequence of the DNA segment, this is a segment of DNA, it has to be transcribed. So, the first point is that it has to be melted, okay. So, melted means a single stranded DNA template has to be generated. So, the generation is done by a kind of molecule or an enzyme called as RNA polymerase. Okay. So, the RNA polymerase is the enzyme which synthesizes all sort of RNA molecules. So, what happens before this the congregation of RNA polymerase or any other your transcription factors in case of eukaryotes or the nucleosomal modifiers, the conglomeration of all this, okay. So, we are not going into this complicacy, we have to understand what is the fundamental of the transcription. So, the first thing is that the exact DNA segment which has to be transcribed, a small portion of that will have to be, or it has to be melted first, okay. So, once the melting is done, this is not like the whole DNA will be melted at one time. So, once the melting is done, now the RNA polymerase, now you can see this is the RNA polymerase enzyme. So, the RNA polymerase enzyme, this acts and it goes on transcribing RNA molecules in the direction of transcription, and here a transcription fork is created. If this is the direction of transcription, so in this direction it will go on synthesizing a new copy or a copy of RNA, okay, and this is called as RNA transcript. Well, But what is that? Now here one thing is that now towards this direction the DNA molecule will be melted till it reaches the end point or the termination point. But in the reverse direction, as the RNA is transcribed, it will come out of the RNA polymerase environment like this, okay, and it will be extended into the cytoplasm or into the nucleoplasmic pool, and towards this direction your DNA again it will be wrapped as a double stranded molecule. Well, so there is a unwinding point, and there is a rewinding point. So the melting of DNA in case of transcription is not a permanent feature, rather it is temporary. For certain time it is open. After the transcription or the transcription of that particular segment is over, now again it will be what? Rewind. So in the direction of transcription, it will go on unwinding the DNA molecule into single stranded template, and in the reverse direction as the transcription is over, okay, now there will be rewinding of DNA molecule, so that the DNA molecule will remain double stranded as such as an intact molecule, and it gives its identity. For a short time it is melted, or it is what it is generated into a single stranded template, basing upon which a RNA transcript or a RNA molecule is formed, okay, or a RNA molecule is synthesized. So another important fact is there. So what is that fact? Now here, now you see, now both of those DNA strands, they are single stranded now at a particular point of time, but the RNA transcript will be formed on a single DNA template or on a single DNA strand, not on both single strands of DNA, okay. So what it makes? There is a sense strand, or this is called as coding strand, okay, and there is an antisense strand, or this is called as nonsense strand, or the most importantly the template strand, okay. There is a coding or sense strand and there is an antisense strand or template strand. The RNA will be synthesized on the template strand only, and the RNA molecules that will be produced here in this process of transcription will be an exact copy of the sense strand or the coding strand. The only difference being, now here all the T molecules will be replaced by U, okay. Why it happens so? Okay. Now again I have come back to the original picture. Now we will again discuss it. Now as I told you, there is a unwinding point and there is a rewinding point. Okay, there is an unwinding point and there is a rewinding point. So what it makes it the sense that now here as a transcription bubble is formed or a transcription fork is formed, okay, which is single stranded in nature. Here the DNA is single stranded, here also the DNA is single stranded, but the RNA transcript or the RNA molecule that has to be synthesized, it has to be synthesized on a single strand of DNA, and that strand of DNA is called as the template strand or the antisense strand. So this strand, where, so this is the strand, you see this blue color line or the upper blue color line is the DNA where the RNA has to be synthesized, and this is called as the template strand or the antisense strand, okay. And the other strand or the other complementary strand of this DNA molecule is called as the sense strand or the coding strand, okay. And the coding strand can never be, or this can never act as a template, Okay. And the RNA molecule, now you compare the RNA molecule or the RNA transcript that is produced here by the help of the RNA polymerase enzyme. Now this will be an exact copy of this coding strand or the sense strand. The only difference being, now here you see the T is replaced by a U. Okay. Wherever there is T, it is being replaced by U. So that is why a RNA transcript which has to be synthesized, now it has to be synthesized upon the template strand or the anti-sense strand. Okay. So this is all about the fundamentals of transcription. Let us go into the details of transcription. Okay. Now again what is transcription? This is a process of synthesis of RNA molecule, and all these necessary informations are there here. And the most important, the enzyme involved in RNA transcription is the RNA polymerase. And unlike DNA polymerase, it does not require any RNA primer or primase enzyme for the initiation of transcription. Okay. Now, what are the steps of transcription? The steps of transcription, as the transcription is an enzymatic process, the mechanism of transcription completes in 3 steps. Okay. 3 major steps, one is initiation, elongation and termination. Now in case of initiation, we will go later on. So let us discuss first, that is formation of a closed complex first, then there is formation of open complex, then ternary complex. How is that? I will explain you. And so what is initiation? So how the process of transcription will begin or how it begins. So it begins at a particular point. That means the transcription, let us suppose there would be a DNA segment, it has to be transcribed into a molecule of RNA, may it be mRNA, tRNA or rRNA. Okay. Now, it does not mean that transcription will begin at any point and it will go bidirectional like DNA replication. Okay. This is not bidirectional, rather unidirectional, and it will not initiate at any point it likes. So there is also a fixed point or a particular point at which the transcription begins, and that point is called as what? Promoter. So the point which is from which your transcription begins, this point is called as promoter, and from the promoter site the RNA molecule bind to the DNA molecule, from the promoter side the RNA polymerase, the enzyme for the synthesis of the RNA molecule, that binds to the DNA molecule and it forms the closed complex. Once the RNA polymerase is successful in melting the DNA strand at that particular promoter site, okay, now it forms an open complex. That means just the binding of RNA polymerase to the DNA molecule at the promoter site, it forms the closed complex. Then after the binding, when it melts the DNA at that particular promoter site to the initiation site, now it forms the open complex. Once melting is done, DNA is open, means now this becomes an open complex. Next, what is the next step? What would be the next step? Now RNA polymerase, what is the function of RNA polymerase? Obviously it has to transcribe the RNA or it has to create a copy of RNA, synthesize an RNA molecule. So when the RNA polymerase synthesizes an RNA transcript or a short strand of RNA, now it forms another complex which is called as tertiary or ternary complex, okay, either tertiary or ternary complex. Now why it is called as tertiary complex? Because the DNA molecule, it is melted, one DNA molecule is involved, then RNA polymerase, this is a protein molecule is involved here, and the RNA molecule which is being produced, that is a nucleic acid which is a ribonucleic acid. 3 molecules are involved, DNA, RNA polymerase and RNA transcript is formed. So because of 3 different molecules, now it is called as a tertiary complex formation or a tertiary complex. Then from once it is being melted and everything is initiated, or the first base is added as per the complementary base pairing between the template strand and the or the new strand of RNA that has to be synthesized. Now it goes on elongating the strand. So elongation is nothing but this is just like what? It is just like the addition of bases one after another, but there is a point called as proofreading, and the proofreading is also being done by the same enzyme called as RNA polymerase. So the elongation is nothing but just the addition of new bases as per the complementary base pairing, the formation of or the elongation of the RNA transcript, or the formation of a long chain of RNA transcript. How much time elongation will take? Till it reaches a termination site or till the process of transcription is over. Okay. So, in this way 3 points or 3 steps are there. One is initiation, that means the transcript formation will be initiated, the RNA transcript. Then elongation, the RNA molecules will go on elongating itself, or the chain will elongate and it will come out of the RNA polymerase environment. And finally the termination. So, at some point it has to terminate, either there is a fixed point termination site or there are some molecules which will be involved in the act of termination. Okay. So, these are the steps. So, before going to the initiation, we should know very much about the promoter region. As we have discussed the origin of replication in case of replication. Okay. So, as there is a fixed point from where your DNA replication starts or is initiated, similarly there is a point from where your RNA transcription begins or it is initiated, and that point is called as promoter. So, let us have a look on the promoter site. So, promoters, they are mostly an upstream region. Okay. So, let me tell you now here, this is a promoter region. Now, what it tells about, now let us suppose this is the DNA molecule that has to be transcribed. Okay. Now, at this point of time, this molecule has to be transcribed. Now, what happens? There is the first point from where the new base or the nitrogenous base, okay, the new base of the RNA molecule, if this is the RNA molecule, when the new base has to be added, and this point is called as +1 initiation point. This is called as +1 initiation point. Okay. And from here onwards, when the next nucleotide will be added or the nitrogenous base, it will be considered as +2, then +3, +4, +5, +6 likewise. Okay. And as this is the direction of transcription or transcription goes in this direction, okay, we will call this as the downstream region. Okay. Whereas whatever nitrogenous bases or the nucleotides will be present in the DNA strand, before the initiation point they will be denoted as -1, -2, -3, -4, -5 and likewise. Okay. And this region are called as what? Upstream region. Okay. And mostly the promoters are present towards the upstream region of a DNA strand. So, they are mostly present in the upstream region. Okay. If we will go on analyzing the promoter of E. coli, okay, the famous model organism where everything is very much established like replication, transcription and translation. So, when it comes to E. coli, now the E. coli has an extended promoter region which is two important reasons. One is a -10 region, as I told you, these are all -10 region, okay, or these are all minus region or the upstream region. So, the upstream regions are denoted as -1, -2, -3, -100 like this, okay. So, this is a -10 region and a -35 region which has some conserved base sequence. Here the sequence is like, this is called as Tata box or something like that. So, the Tata box has TA, TA, AT sequence, okay, and the minus 35 resin it also has a conserved sequence like TTGACG. And what has to be done here, this promoter region, now as you can see by this color of this promoter region, this is from this to this point, this is the double stranded DNA and this is the extended promoter region. This is the +1 initiation site. That means the fast base of RNA will be added at this point, okay. Let us suppose there would be A here. So, what will be added here? A U will be added here as the first base of the RNA molecule, okay. So, the promoter is extended from +1 to -35 resin. It has two conserved sequence, -10 resin and -35 resin. So, what happens here? Now, as you can see some grey portions or this grey molecule, this is the RNA polymerase molecule, this portion, okay. So, this is the RNA polymerase molecule. This extensively it binds to this region from the -35 region towards the downstream region, okay. It would be like +10 somewhat upwards like this. So, the RNA polymerase extensively binds to this region and it melted the structure here, okay. So, that the fast base will be added by the RNA polymerase molecule and this RNA polymerase molecule will have a catalytic site over to add the fast base and later on it goes on in this direction forming the RNA transcript, okay. So, this is the promoter region where, so what is this region known as? Promoter region is the region where the RNA polymerase molecule binds and it starts or it initiates the transcription. So, how the initiation of transcription begins? Now, in case of bacteria, there is a single RNA polymerase molecule and this single RNA polymerase molecule has many components or many subunits like alpha, beta, beta dash, omega subunits and there is a sigma factor and this sigma factor here you can see the sigma factor of the RNA polymerase that actually facilitates or that actually helps in recognizing this promoter site and it binds to there and once the initiation begins, now this sigma factor it falls off, okay. And these are the subunits of the RNA polymerase. Now, there is a single RNA polymerase molecule in case of prokaryotes, whereas in case of eukaryotes, there are 3 RNA polymerase molecules or polymerase enzyme, okay. So, the core polymerase or the polymerase without the sigma factor, this is called as core polymerase and the core polymerase plus the sigma factor, they togetherly they constitute the holoenzyme. So, the core polymerase with sigma factor is called the holoenzyme and in case of E. coli, it consists of 2 conserved sequence as we already have discussed, one as a -35 element, another one is at the -10 element. So, we already have discussed this. So, what happens as I told you, first it forms a closed complex, that means the RNA polymerase binds to the DNA, simply the DNA is closed, so that is why it is called as a closed complex, then open complex as the RNA polymerase it mails the DNA, now it becomes an open complex. Once the RNA molecule or the RNA polymerase transcribe a short RNA molecule or a short RNA transcript, now this becomes a tertiary complex, okay. But in case of this, now many times the RNA transcript is formed and it is aborted, that means it is discarded. So, once it overcomes a strength of or once it overcomes a length of 10 nucleotides long RNA transcript, now it goes on elongating the chain towards the direction of transcription. So, this is the picture where you can see this is the promoter region where the RNA polymerase has bind with along with the sigma factor. Now you see it has opened up the structure, now it becomes an open complex, well, and when it forms a SOT molecule, now as the initiation site is here, so once it added the first base, it becomes a tertiary complex. Okay. So, next elongation, elongation is nothing but just the addition. Now, you see it is nothing, but once it is initiated, it goes on elongating. Now, you see, now here it has initiated here, then you see now it has been extended up to this by creating a long RNA molecule which has come out of this RNA polymerase enzyme. Okay. And it goes on what elongating the RNA transcript till it reaches a terminator. Okay. Or the terminator can be a site, the terminator can be an exogenous molecule. Okay. So, this is nothing but the most important thing is that it always goes for a proofreading. That means, now in this case no wrong base will be added to as per the self or complementary base pairing, no wrong base will be added. Once it adds a new base or new nucleotide to the elongating RNA transcript or to the elongating RNA chain, now it again what comes or it retracts back and it takes a proofreading whether I have added the exact or the right nucleotide or not, it checks it about. Okay. Next, the most important is the termination. Termination is very important. Now, termination in case of prokaryotes, it happens in two ways. One is a row independent and another one is a row dependent termination. So, first let us have a look on the row independent termination. Now, in case of row independent termination, actually when the RNA transcripts it goes on elongating it goes on elongating till it reaches a site called as terminator site. In case of E. coli, this terminator site is a self complementary GC rich region. So, once that GC rich region is transcribed into the RNA segment or it is transcribed to the RNA transcript, now after it slides over the rest of the DNA segment, now it forms a self complementary region and it forms a stem region and a loop region, as you can see it like this. Okay. So, when what happens here, as you can see, now this is an extended region where this is the GC rich self complementary region. So, once the RNA transcript it transcribed that GC rich self complementary region, now it forms a loop like this, a stem, this is GC rich, now you see GC, CG, GC like this, okay, having a single stranded region also, this is, so this forms a stem and a loop, okay. So, after that there are a what in the template strand, the GC rich self complementary region is followed by a poly A nucleotide long chain of the DNA. So, when it is transcribed into the RNA chain, now the same GC rich region comes to this point along with many U regions. Okay. So, the A is transcribed into U, okay, on the RNA transcript. So, as it forms a self complementary GC rich stem and loop region followed by U, which forms a RNA DNA hybrid, okay, with the DNA is the template and RNA is formed nascent. So, this U, many U and many A base pairing is so weak, this is so weak that it dissolves or it breaks the chain and the total RNA transcript, it falls out of the RNA polymerase environment and everything is dissociated. So, this is the reason you can see this is the stem, this is the loop region and it is followed by U, so that the RNA polymerase cannot go further and this what it exerts a pressure that pulls out of the RNA transcript from the RNA polymerase and everything get dissociates from the DNA molecule. Now, the DNA is again double stranded and your RNA transcripts comes out. Okay. So, this is the row independent kind of termination. So, next is the row dependent termination. In case of row dependent termination, there would be no such termination site. Okay. Rather, in case of row dependent, there are certain row proteins. Now, here there is no site in the DNA molecule, rather some group of proteins. Now, these are called as row proteins, well, and they binds to the nascent chain of the RNA molecule. Well, once they binds, now they slowly chase the movement of transcription. So, as the movement is faster, when they start increasing their movement, this row protein slowly comes to the RNA polymerase at this point, and finally it forms an interaction. It has a RNA DNA helicase activity. Okay. So, what it happens, it breaks the hydrogen bonds that is present between the DNA strand and the RNA transcript. Okay. So, this row proteins, now you see this has a RNA DNA helicase activity. So, once it reaches to the RNA polymerase molecule or into the RNA polymerase, now it dissociates everything and you get your RNA transcript. Okay. And in this way termination happens. So, one is row independent, another one is row dependent. Now, you see in case of row independent, because of the pressure exerted by the RNA transcript itself, because of the transcription of the G series self complementary region followed by poly U, it cannot support the RNA transcript or the DNA RNA hybrid, and it pulls the structure from the RNA polymerase and everything dissociates. Well, and here in case of row protein, it changes the transcription machinery and arrests the transcriptional machinery, and it pulls out the RNA transcript and everything is dissociated, and in this way termination occurs in case of row dependent manner. Okay. So, this is all about initiation, elongation and termination of transcription. But after transcription, now what we are going to get? We are going to get either an mRNA or a tRNA or a rRNA. Okay. So, out of this, whatever RNA we are going to get, mostly everyone will undergo a post transcriptional modification, but we will stick to the mRNA, because in case of mRNA it undergoes the process of translation to form a protein molecule. So, what happens here? Now, the post transcriptional modification, now here we are focusing on the post transcriptional modification of mRNA. Now, mRNA in case of prokaryotes, it certainly undergoes a very little post transcriptional modification, or it does not undergo any, mostly hardly it takes any kind of modification. So, as it it comes out of the RNA polymerase environment, now these ribosomes, they get attached to the nascent RNA and this starts synthesizing the protein molecules in case of prokaryotes. But in case of eukaryotes, once the RNA transcript is fully formed and mostly the mRNA, now it undergoes many processes like splicing, 5' capping and 3' poly A tail addition. Okay. So, what it means? So, what splicing means? Now, splicing means, unlike prokaryotes, now there is no such you can say as any kind of junk DNA. Okay. So, the sequence of DNA which is or the part of gene, it is continuous. So, there is no interrupted regions, but in case of eukaryotes, now there are certain regions, as you can see the black here, these regions, they are the interrupted regions which actually which actually is not the part of the mRNA that has to be translated. So, this part has to be cut off. Well, so you see the DNA, it transcribes the RNA, the same interrupted region, these are called as introns. Okay, this is intron 1, intron 2, intron 3 and intron 4. So, these regions are transcribed into the RNA strand, particularly to the mRNA strand, and these introns has to be cut off. So, here you see these regions are the exonic regions, exon 1, exon 2, 3, 4 and 5, and these blue regions, okay, these regions, it has to be cut off. And this cutting of these intronic regions, this is called as splicing. Okay. So, once these regions are cut, all these regions, exonic regions, they are joined together to form the coding mRNA region. Well, and this molecule is actually get translated into a protein. So, splicing is the cutting of the non-coding region of the DNA after it has been transcribed into the mRNA. So, before the mRNA undergoes the process of translation, this intronic region which has been transcribed into the mRNA strand, they has to be cut out. Well, and after the cutting, all the coding region, they are joined together to form a continuous chain or to form a continuous segment which actually helps in translation. So, this is known as splicing. Then after that, now the mRNA molecule, it has to be protected in case of eukaryotes. So, a 5 prime cap is added to the mRNA towards the 5 prime site, and a 3 prime towards the 3 prime end, a poly A tail, that means many A, like 100 A's or 200 A's like this, or even 1000 A's. So, the poly A tails are added like this. Okay. These are all post-transcriptional modifications. These are all post-transcriptional modification. Besides that, tRNA molecule, they also undergoes post-transcriptional modification, but we are much concerned about mRNA, as mRNA only undergoes, or mRNA only act as the template for the synthesis of protein as per the genetic code or as per the codon. So, one codon, it acts or it codes for a single amino acid, and this amino acid, or as per the codons, well, now the amino acids are joined together to form a long chain of polypeptide which undergoes certain modification to form a functional polypeptide or a functional protein. Well, so and proteins, they have several functions, that is innumerous. So, we will, while we are discussing translation, we will discuss. So, this is all about the mRNA processing. Now, there is the splicing, the cutting of the intronic region, the addition of a 5 prime cap, and mostly this is a guanine nucleotide that is added towards the 5 prime end, and the addition of 3 prime poly A tail. This is all about the post-translational modification of mRNA in case of eukaryotes. Rather, prokaryotes or the prokaryotic mRNA, it does not undergo any sort of your post-transcriptional modification. Okay. So, this is all about transcription and this post-transcriptional modification. Before ending off, let me tell you one fact. Just let me tell you. Yes. So, one more fact is that in case of prokaryotes, a single RNA polymerase is transcribes all the RNA, may it be mRNA, tRNA or rRNA. Okay. But in case of eukaryotes, 3 RNA polymerase enzymes are there which transcribes now like RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. Now, RNA polymerase 2 transcribe all sort of mRNA that codes for protein, whereas RNA polymerase 1, it transcribes for all rRNA molecules along with some small nucleolar RNAs. Whereas RNA polymerase 3, it transcribes mostly all the tRNA molecules. Well, and sorry, the small nucleolar, the small nucleolar RNAs, these are being transcribed by the polymerase 3 molecule or polymerase 3 enzyme, RNA polymerase 3 enzyme. So, here you can see there is a single RNA polymerase in case of prokaryotes, whereas in case of eukaryotes there are 3 RNA polymerase to transcribe 3 different RNAs. Polymerase 2 transcribes mRNA, polymerase 1 transcribes rRNA, polymerase 3 transcribes tRNAs. Okay. And besides the RNA polymerase, lots of other factors or proteins are involved, like many transcription factors, and what about nucleosome modifiers. Okay, they are also involved along with the RNA polymerase molecule in case of eukaryotes. So, this is all about transcription. Okay. Now, this is all about transcription and post-transcriptional modifications like splicing, mRNA processing like for 5' capping and 3' poly A tail addition, and finally thank you. In the next class we will go for another new chapter. Have a good day.